Hello friends, and welcome to our today's lecture on overview of phages, viroids, mycoplasma and E. coli. In today's lecture, we will discuss about bacteriophages, their history, classification and genome structure, characteristic features of viroids, history, taxonomy, pathogenesis and origin, general overview of mycoplasma, and in the end we will talk about the structure and function of E. coli and its role in biotechnology. At the outset, let us talk about bacteriophages. A bacteriophage, informally phage, is a virus that infects and replicates within a bacterium. Bacteriophages are composed of proteins that encapsulate a DNA or RNA genome and may have relatively simple or elaborate structures. It infects bacteria by inserting its genetic material into the host cell. Now the history. French-Canadian microbiologist Felix de Hellery, working at the Pasteur Institute in Paris on 3rd September 1917, discovered an invisible antagonistic microbe of the dysentery bacillus. De Hellery called the virus a bacteriophage or bacteria eater. As far as the classification of phages is concerned, they occur abundantly in the biosphere with different virons, genomes and lifestyles. Phages are classified by the International Committee on Taxonomy of Viruses according to morphology and presence of nucleic acids. 19 families are currently recognized by the ICTV that infect bacteria and archaea. Of these, only 2 families have RNA genomes and only 5 families are enveloped. Of the viral families with DNA genomes, only 2 have single-stranded genomes. 8 of the viral families with DNA genomes have circular genomes, while 9 have linear genomes. 9 families infect bacteria only, 9 infect archaea only, and 1 Tactiviridae infects both bacteria and archaea. Now we will discuss the replication of bacteriophages. They may have a lytic cycle or a lysogenic cycle, and a few viruses are capable of carrying out both. With lytic phages such as the T4 phage, bacterial cells are broken up, lysed and destroyed after immediate replication of the virion. Some lytic phages undergo a phenomenon known as Lysis Inhibition, where completed phage progeny will not immediately lyse out of the cell if extracellular phage concentrations are high. In contrast, the lysogenic cycle does not result in immediate lysing of the host cell. Those phages able to undergo lysogeny are known as temperate fades. Their viral genome will integrate with host DNA and replicate along with it fairly harmlessly, or may even become established as a plasmid. Some prophages may provide benefits to the host bacterium, while they are dormant by adding new functions to the bacterial genome in a phenomenon called lysogenic conversion. Example are the conversion of harmless strains of Corynebacterium diphtheria or Vibrio cholera by bacteriophages to highly virulent ones which causes diphtheria or cholera respectively. Now the virion assembly. In the case of the T4 phage, the construction of new virus particles involves the assistance of helper proteins. The base plates are assembled first, with the tails being built upon them afterwards. The head, capsids, constructed separately, will spontaneously assemble with the tails. The DNA is packed efficiently within the heads. The whole process takes about 15 minutes. Release of virions. Phages may be released via cell lysis, by extrusion, or in a few cases by adding lysis. By tailored phages is achieved by an enzyme called endolysin, which attacks and breaks down the cell wall peptidoglycan. Now the genome structure of bacteriophages. Bacteriophage genomes are especially mosaic. The genome of any one phage species appears to be composed of numerous individual modules. These modules may be found in other phage species in different arrangements. Mycobacteriophages. Bacteriophages with mycobacterial hosts have provided excellent examples of this mosaicism. Now we will discuss briefly about viroids. Viroids are the smallest infectious pathogens known, consisting solely of short strands of circular single-stranded RNA without protein coats. They are mostly plant pathogens, some of which are of economical importance. Viroid genomes are extremely small in size, ranging from 246 to 467 nucleobases. In comparison, the genome of the smallest known viruses capable of causing an infection by themselves are around 2000 nucleobases in size. The human pathogen hepatitis D virus, a defective RNA virus, are similar to viroids. The first viroid to be identified was potato spindle tuber viroid. Some 33 species have been so far identified. Viroids do not code for any protein. Viroids' replication mechanism use RNA polymerase II, a host cell enzyme normally associated with synthesis of messenger RNA from DNA, which instead catalyzes rolling circle synthesis of new RNA using the viroid's RNA as template. Now we will talk about the history of viroids. In 1920s, symptoms of a previously unknown potato disease were noticed in farmers' New York and New Jersey fields. Because tubers on affected plants become elongated and misshaped, they named it the potato spindle tuber disease. Viroids, the first known representatives of a new domain of subviral pathogens, were discovered initially characterized and named by Theodore Otto Dinier in 1971. Later, Sanger 1976 presented evidence that potato spindle tuber viroid is a single-stranded, covalently closed circular RNA molecule existing as a highly circular RNA, unlike linear RNA, forms a covalently closed continuous loop in which the 3' and 5' ends present in the linear RNA molecules have been joined together. Now the taxonomy. There are two major families of viroids, family Pospiviroideae, family Evsonviroideae. Transmission. Viroid infections are transmitted by cross-contamination following mechanical damage to plants as a result of horticultural or agricultural practices. Some are transmitted by aphids, as they can also be transferred from plant to plant by leaf contact. Now the replication. Viroids replicate in the nucleus as in Pospiviroideae or in chloroplast like Evsonviroideae or plant cells in 3 steps through an RNA-based mechanism. They require RNA polymerase II, a host cell enzyme normally associated with synthesis of messenger RNA from DNA, which instead catalyzes rolling circle synthesis of new RNA using the viroid as template. Now the viroids and RNA silencing. There has long been uncertainty over how viroids are able to induce symptoms in plants without encoding any protein products within their sequences. Evidence now suggests that RNA silencing is involved in the process. Living relics of the RNA world, Dinier's 1989 hypothesis proposed that unique properties of the viroids make them more plausible macromolecules than introns or other RNAs considered in the past as possible living relics of a hypothetical pre-cellular RNA world. These properties are viroid small size imposed by error-prone replication. They are high GC content, which increases stability and replication fidelity. Their circular structure, which assures complete replication without genome tags. Existence of structural periodicity, which permits modular assembly into enlarged genomes. Their lack of protein coding ability, consistent with a ribosome-free habitat and replication mediated in some by ribozymes. Now the pathogenesis of viroids. Probably there is more than one mechanism responsible for viroid pathogenesis. One pathway is due to viroid RNA activating a plant RNA protein kinase or PKR that is analogous to the PKR enzyme activated by viral RNAs in mammalian cells. Protein synthesis is reduced and this causes pathogenic effects. Viroids, the smallest known pathogens, are naked, circular, single-stranded RNA molecules that do not encode protein yet replicate autonomously when introduced into host plants. Two economically important viroids are coconut cadang viroid, which causes a lethal infection of coconut palms, and apple scar skin viroid, which causes an infection that results in visually unappealing apples. Symptoms of viroid infection in plants include stunting of growth, deformation of leaves and fruit, stem, necrosis and death. Now dear friends, we will give a brief account on Mycoplasma. Mycoplasma is a genus of a bacteria that lack a cell wall around their cell membrane. Without a cell wall, they are unaffected by many common antibiotics such as penicillin or other beta-lactam antibiotics that target cell wall synthesis. They can be parasitic or saprophytic. Several species of pathogenic in humans include Mycoplasma pneumoniae, which is an important cause of a typical pneumonia and other respiratory disorders, and Mycoplasma genitalium, which is believed to be involved in pelvic inflammatory disease. Mycoplasma species are the smallest bacterial cells, can survive without oxygen and are typically about 0.1 micrometer in diameter. The term mycoplasma, from the Greek myx, fungus, and plasma formed, was first used by Albert Bernhard Frank in 1889. Julian Novak later proposed the genus name Mycoplasma for certain filamentous microorganisms. Some of the species that infect humans are Mycoplasma amphoiforme, Mycoplasma buccali, Mycoplasma faecium, Mycoplasma fermentans, Mycoplasma genitalium, etc. and many more. Now the pathophysiology of Mycoplasma species. Mycoplasma species have been isolated from women with bacterial vaginosis. Mycoplasma genitalium infection is associated with increased risk of cervicitis, pelvic inflammatory disease, preterm birth and spontaneous abortion, and infertility. Mycoplasma are associated with fetal respiratory distress syndrome, bronchopulmonary dysplasia and intraventricular hemorrhage in preterm infants. Over 100 species have been included in the genus Mycoplasma. Mollicutes or parasites or commensals of humans, animals and plants. Now the taxonomy of Mycoplasma species. Mycoplasma cells are physically small, less than 1 micrometer, so are difficult to detect with a conventional microscope. Mycoplasma may induce cellular changes including chromosome aberrations, changes in metabolism and cell growth. Severe Mycoplasma infections may destroy a cell line. An estimated 11 to 15% of US laboratory cell cultures are contaminated with Mycoplasma. Now the pathogenicity. The P1 antigen is the primary virulence factor of mycobacteria. P1 is a membrane-associated protein that allows adhesion to epithelial cells. Several Mycoplasma species can cause disease, including Mycoplasma pneumoniae, which is an important cause of a typical pneumonia, and a Mycoplasma genitalium, which is associated with pelvic inflammatory disease. Mycoplasma infections in humans are associated with skin eruptions in 17% of cases. Sexually transmitted infections like Mycoplasma and Ureaplasma species are not part of the normal vaginal flora but are separate through sexual contact. Infertility. Some Mycoplasma may have negative effect on fertility. Infant mortality, low birth weight, preterm infants are susceptible to Mycoplasma infections. Links to the cancer. Several species of Mycoplasma are frequently detected in different types of cancer cells. These species are Mycoplasma fermentans, Mycoplasma genitalium, Mycoplasma hyaluronus, Mycoplasma penetrans. The majority of these mycoplasmae have shown a strong correlation to Malaysian transformation in mammalian cells in vitro. Possible intracellular mechanism of mycoplasmal malignant transformations. Cells infected with mycoplasma for an extended period of time show significant chromosomal abnormalities. These include addition of chromosome, loss of entire chromosome, partial loss of chromosome, and chromosomal translocation. All of these genetic abnormalities may contribute to the process of malignant transformation. Chromosomal translocation and extra chromosomes help create abnormally high activity of certain proto-oncogenes. The activity of proto-oncogenes is not the only cellular function that is affected. Tumor suppressor genes are affected by chromosomal changes induced by mycoplasma as well. Partial or complete loss of chromosome causes the loss of important genes involved in the regulation of cell proliferation. Another possible mechanism of carcinogenesis is RAC1 activation by a small GTPase like protein fragment of mycoplasma. Partial reversibility of malignant transformation. The malignant transformation induced by mycoplasma is also different from that caused by other pathogens in that the process is reversible. The state of reversal is, however, only possible up to a certain point during the infection. In the case of Mycoplasma fermentans, the transformation is reversible until around week 11 of infection and starts to become irreversible between weeks 11 and 18. If the bacteria are killed using antibiotics like ciprofloxacin or clarithromycin before the irreversible stage, the infected cells should return to normal. Types of cancers associated with mycoplasma: colon cancer, gastric cancer, lung cancer, prostate cancer, renal cancer. Mycoplasma pneumonia is spread through respiratory droplet transmission. Once attached to the mucosa of a host organism, Mycoplasma pneumoniae extracts nutrients, grows and reproduces by binary fission. Attachment sites include the upper and lower respiratory tract, causing pharyngitis, bronchitis and pneumonia. Chronic mycoplasma infections have been implicated in pathogenesis of rheumatoid arthritis and other rheumatological disease. Mycoplasma atypical pneumonia can be complicated by Stevens-Johnson syndrome, hemolytic anemia, cardiovascular disease, encephalitis or Guillain-Barré syndrome. So far we have talked about phages, viroids and mycoplasma. Now we will talk about description and significance of E. coli. E. coli was first discovered in 1885 by Theodor S. Church, a German bacteriologist. E. coli is a faculty to aerobic and anaerobic growth, gram-negative rod-shaped bacteria that can be commonly found in animal feces, lower intestines of mammals, and even on the edge of hot springs, and grows best at 37 degrees centigrade. E. coli is a gram-negative organism that cannot spirulate. Therefore, it is easy to eradicate by simple boiling or basic sterilization. E. coli can also be classified into hundreds of strains on the basis of different serotypes. Furthermore, enteric E. coli can be classified into 6 categories based on its virulence properties: enterotoxigenic E. coli, enteropathogenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, enteroadherent aggravative E. coli, and virotoxigenic E. coli. Most E. coli live in our intestines, are part of the normal flora of the gut, and can benefit their hosts by producing vitamin K2 and prevent colonization of the intestine with pathogenic bacteria. Now, the genome structure of E. coli. E. coli has one circular chromosome with about 4600 kilobase, about 4300 potential coding sequences, and only about 1800 known E. coli proteins. Cell structure. E. coli is a gram-negative rod-shaped bacteria which possess adhesive membrane and a cell wall that consists of an outer membrane containing lipopolysaccharides, a periplasmic space with a peptidoglycan layer, and an inner cytoplasmic membrane. Ecology of the E. coli. E. coli can be commonly found in lower intestine of human and mammals. When E. coli locates in human large intestine, it can help digestion process, food breakdown, and absorption and vitamin K production. E. coli can also be found in environments at higher temperature, such as on the edge of hot springs. E. coli is commonly used as an indicator in the field of water purification. E. coli index can indicate how much human feces is in the water. Now the pathology. Some strains of E. coli can cause severe infections in many animals such as human, sheep, horses, dogs, etc. The one that only found in humans is called enteroaggregative E. coli. Urinary tract infection, for example, can be caused by the ascending the infections of urethra. E. coli 0157H7 is one of the most infective strains that can cause food poisoning and can lead to bloody diarrhea and kidney failure when one gets infected by contaminated ground beef, unpasteurized milk, or contaminated water. The individual may experience the following symptoms. First, abdominal pain, diarrhea, nausea, vomiting, fever, fatigue. Causes of E. coli infection. Most strains of E. coli are harmless. However, one group, E. coli 0157H7, produces a potent toxin, schizotoxin, that is harmful for the lining of the small intestine. Humans can become infected by ingesting contaminated water and food, having physical contact with animals, and high fiber diets. Now the treatment. The mainstay of treatment is the assessment of dehydration and replacement of fluid and electrolytes. Administration of antibiotics has been shown to shorten the course of illness. Oral rifaximin, a semisynthetic rifamycin derivative, is an effective and well-tolerated antibacterial for the management of adults with non-invasive travelers' diarrhea. Now the prevention methods. Besides treatment, there are some important preventive measures as well. Wash hands before and after cooking. Avoid cross-contamination of foods. Keep food refrigerated or frozen. Make sure food is kept at appropriate temperature. Avoid raw meat and poultry. Don't let food defrost on counter. Now the application of E. coli in biotechnology. E. coli plays an important role in current biological engineering because of its manipulation and long laboratory history. Widely used to synthesize DNA and proteins. The most useful contribution of recombinant DNA from E. coli is to use the manipulation of E. coli to produce human insulin for diabetes patients. Plays an important role to enable the rapid, continuous production and purification in large amount of human A-beta sequence by its unique expression system to cure Alzheimer's disease. With this, we conclude today's lecture on overview of phages, viroids, mycoplasma and E. coli. Thank you and hope you enjoyed.